Let me welcome you to this lecture. In the last lecture, we have learned about the oxidative addition reaction as well as the reductive elimination reaction, and during this lecture, we will learn about another class of important reactions in organometallic chemistry. Now, what we have learned in oxidative addition reaction is that we start with a precursor metal complex and then it undergoes oxidative addition, that is two anionic ligands are added to the metal center so that the metal changes its oxidation state by 2 units as well as its coordination number also increases by 2 because both of these two ligand fragments, that is X and Y, are now coordinated to the metal center. So we can visualize this reaction as an insertion of this metal onto this bone between X and Y. So only thing here is that along with the insertion also the oxidation number of the metal changes. So when the oxidation number of the metal does not change in such a reaction, so we term such reaction as insertion reaction. And again, in organometallic chemistry, in organometallic catalysis, there are many examples of insertion reaction in important catalytic cycles, and we will learn some of these reactions. And during this lecture, we will primarily focus on two kinds of insertion reaction. First is insertion of carbon monoxide into a metal carbon bond. And the second reaction that we will look at is insertion of ethylene in a MH bond. So these are the two reactions that we will discuss over here. Now let us go and look at a classic example of the first type kind of reaction, that is insertion of carbon monoxide in a MC bond. This is manganese carbonyl complex. If one starts with, and do reaction with carbon monoxide, the product that we obtained is where carbon monoxide gets inserted into this metal to carbon, that is CH3 bond, and the product that we obtained is this. So what we initially can predict is that in this reaction, carbon monoxide essentially gets inserted into this metal to carbon bond, and that is why we call it insertion reaction. But then when we look at the reaction carefully, or when we look at the mechanism of the reaction, what we find is that instead of this carbon monoxide getting inserted into this manganese methyl carbon bond over here, it is the methyl group which migrates into one of the carbonyl ligand, that is intramolecular carbonyl ligand, that is already present in the complex, and that is how a vacuum coordination site is essentially produced here, and the carbon monoxide ligand comes and attack on the metal to give us this resulting complex. So what we will do here is that we will look at both of these two possibilities, that is insertion of carbon monoxide into this metal carbon bond, as well as we will see how the methyl group migrates to one of the carbonyl ligand to give us this product. Both of these two mechanisms we will look at. So let's write this as the methyl group here, methyl, and this is the precursor complex over here. Now if one of the carbonyl ligand that is present in the complex itself has to first either this goes and gets inserted here, or for the incoming carbonyl ligand to come and for this reactant carbonyl ligand, that is carbonyl reagent that we have added over here to come and attack. At the moment we do not have any vacant coordination site, so first one of the ligand has to leave, and then only this can come and attack on this to give us the product. So what we look at here is one of the intramolecular carbonyl ligand coming and attack on this MC bond over here, and that will give us the product is going to be now so what we will have here is now a vacant coordination site, and the reactant carbon monoxide can now come and occupy this place. So this is how reaction is going to proceed if it goes via insertion of or CO insertion mechanism. Now let us look at the other possibility, this migration of the methyl group. So methyl migration to one of the carbon monoxide. So then this can come and migrate to one of the carbonyl ligand over here, and in that case what we will end up with is say this sort of a product. It can go to any one of these cis carbonyl. It can migrate to any one of these 4 cis carbonyl ligands in the complex, and what we will end up with is now a vacant coordination site at this place where the methyl group is present. So this is how we can distinguish between carbonyl insertion and methyl migration, and now we will look at mechanistic evidences which suggest us that it is during this reaction, instead of carbonyl insertion, actually methyl migration takes place. So for this, the reaction was carried out using tagged carbon monoxide, that is instead of CO we use the tagged CO, that is 13 carbon CO, and the product in this case is actually so we obtain the cis CO4 13 CO manganese and this acetyl. This as the 100% exclusive product. And no other product is obtained. When we say cis, actually it is cis with respect to the acetyl group over here. So we can draw here maybe. Let's write it here. So we can write 13CO here, or any one of these four places. When we write, then it is cis with respect to this acetyl group, and that is why we call this isomer as the cis isomer over here. And what we have found here is that during this reaction, the cis product is formed as a 100% exclusive product, and no other product is obtained. And also one more thing to note over here is that the acetyl carbonyl group does not have 13 carbon atom over here. So that means this 13CO does not come, this intermolecular insertion reaction does not take place. That is, there is no reaction between this 13CO and this manganese carbon bond over here, or had it happened, we would have obtained some product with the 13CO tagged group, 13 carbon tagged acetyl group over in case of this product. So that does not happen. None of this product contains 13C carbon in this acetyl group, and that essentially confirms us that no intermolecular CO insertion reaction. So this is what it essentially tells us. And also, since only the cis product is obtained, since only the cis isomer is obtained, it essentially tells us that after the product is formed, the CO groups in the product does not scramble, and then gives us a statistical distribution of both cis and trans. So no scrambling. Once the metal to carbonyl bonds are formed, they are stable, they do not break down and then reform, so that a statistical distribution of both cis and trans can form. So no scrambling of CO group in the product to give cis and trans isomers. So this gives a lot of important mechanistic detail about this reaction. Now what further mechanistic details about this reaction can be obtained by studying the reverse reaction, that is coming back from the product to this side of this reaction. It is a reverse reaction, and we can term this reaction as decarbonylation reaction, because one of the carbonyl ligand is essentially lost, and that is also we can term this reaction as an elimination reaction. While the forward reaction we term it as insertion reaction, the backward reaction can be termed as elimination reaction. So we can obtain further mechanistic detail of this reaction by studying the elimination reaction in this 13CO tagged carbonyl complex, which is the product in case of the insertion reaction. So let us go and look at the experimental evidences that we obtained by studying the decarbonylation reaction, because the mechanism of the opposite reaction or the reverse reaction will be exactly opposite to the mechanism of the forward reaction, that is the insertion reaction. So based on this, what we can do is that we can we can predict the mechanism of the insertion reaction by studying the mechanism of the elimination reaction. So what was done is that the decarbonylation reaction was studied. Decarbonylation reaction of the 13 CO tagged manganese acetyl complex, not the methyl complex. That is the product in the insertion reaction was studied. Now, if the decarbonylation happens through the reverse of CO insertion reaction, so that is decarbonylation by reverse of CO insertion. So that is, if we assume that the the forward reaction, that is the insertion reaction, takes place by CO insertion instead of methyl migration, then the decarbonylation reaction will proceed by the reverse of CO insertion reaction. And in that case, what is going to happen is that the CO group in this acetyl ligand will essentially replace any one of these cis carbon monoxide ligands in the starting precursor over here, and that will essentially give us two types of products here, because what is going to happen is the CO group in this acetyl ligand can replace any one of these four carbon monoxide. Now there is one tagged CO group, while three of the CO group are untagged over here. So since there is 1/4 possibility of removing or replacing this tagged carbon monoxide group by the CO group in the acetyl ligand over here, so that is why 25% of the product that is formed in this reaction will not have any tagged CO group. Because there is 1/4 possibility of this CO group, that is the tagged CO group, getting replaced. That is why what we can predict is that 25% of the product will not have any tagged CO group, and while 75% of the product will have the tagged CO group intact. Because in this case there is 3/4 possibility. There are 3 carbon monoxide groups out of the 4 are untagged, and there is 3/4 possibility, 75% possibility of this carbonyl ligand replacing any one of these 3 carbon monoxide groups, and that is how we can obtain 75% of the product, or we can predict that 75% of the product will have a tagged CO group intact. So this is what is predicted if the reaction proceeds by CO insertion reaction. But the reaction, when we study the products obtained in the reaction, what we see is something different apart from this. So what we obtain in case of the product, instead of what we see is that sorry, let's write this CO. So 25% of the product does not have any tagged CO. This is the experimental result, experimentally observed result. This is what is the predicted result if the reaction goes by CO insertion reaction. But the experimental results tell us that yes, 25% of the product does not contain any tagged CO group. But then we do not exactly have 75% of this product, but it's a mixture of two different components. That is another 25%. Let's write this CH3 group at this place. This is where the 13 CO group is intact. So we get another 25% of the product where there is 13 CO group intact, as well as the CH3 group is trans to this tagged 13 CO group, trans isomer. And then finally we have another 50% component where the methyl group is cis to the tagged 13CO group over here. So this is 50% cis isomer is formed, 25% trans isomer, that is the methyl group is opposite to the 13CO group, while 25% of the product does not have any tagged CO carbon monoxide group. So what does it tell us? That the reaction does not proceed by CO insertion reaction but by methyl migration reaction. How these kind of products can form, we can by when we look at the reaction to proceed by methyl migration, we will realize that now instead of carbon monoxide insertion, if the methyl group migrate to any one of these carbonyl ligand, that is cis to the acetyl group over here, so then there is again 4 CO groups are available, cis to the acetyl group, so methyl can migrate to any one of these 4 carbon monoxide groups. So there is 25% possibility of replacing this particular tagged carbon monoxide group, and that is why we obtain 25% of the product where there is no tagged carbon monoxide group, because the methyl group has now replaced this particular CO group. Or then what we have is that 25% we obtained are trans isomer, that is the methyl group is trans to the 13 CO group. So what we see is that there is one CO group over here which is trans to this 13 CO group. Out of these 3 CO groups, there is only one CO group which is trans to this 13 CO group, and when this CO is replaced by the methyl group over here, then we obtained this trans isomer, and that is why we obtain 25% product as the trans isomer. And then what we have is there are 2 CO groups which are cis to this 13 CO group, and that is why 50% of the product are obtained as cis isomer. So these results essentially gives us strong evidence that this decarbonylation reaction proceeds by the reverse of methyl migration. So reverse of methyl migration, and that also tells us that the insertion reaction takes place by methyl migration, not by CO insertion reaction, because the mechanism of the reverse reaction has to be exactly opposite to that of the forward reaction. And in fact, when chemists in organometallic chemistry talk about insertion reaction, we mean about alkyl migration reaction, not insertion of the ligand. Here we mean that alkyl migration. For all practical purposes, insertion reaction essentially means alkyl migration reaction. Now let us look at another one of these insertion reaction, where we again obtain the evidence of alkyl migration instead of CO insertion reaction. In this case, one starts with a chiral complex. So again, if the reaction takes place by CO insertion, this is the chiral tetrahedral iron complex, and if the reaction takes place by CO insertion reaction takes place, then what is going to happen in this case is the CO group will migrate to this metal carbon bond over here, and we will end up with while a vacant coordination site is going to be produced at this place over here, and now the 13CO will come and attack on this vacant coordination site, and that will give us the product like this over here, where the configuration of the chiral center is essentially retained. So we obtain product with retention of configuration. But if the reaction takes place by alkyl migration instead of CO insertion, then the product is going to be different. Now the vacant coordination site is going to be produced where the ethyl group was present, and when the tagged carbonyl group come and attack, now it will come to this place. So now inversion of configuration has taken place. When the reaction took place through alkyl migration. So what was observed is that when the reaction was carried out in nitromethane, the product obtained had inversion of configuration, and only the inverted configuration product is obtained exclusively. So this essentially tells us that the reaction proceeds via alkyl migration mechanism. But then in certain solvents, again we obtain both the products, that is the products with retention of configuration as well as product with inversion of configuration. This tells that maybe both the mechanisms, that is CO insertion and alkyl migration, are operative in such solvents, as well as what can also happen is that the the configuration, the chirality may have been lost during the reaction, and that is how when the product is formed essentially, finally then the products with both the configuration are formed, and that is how we obtain both the products with both the configuration. So there is one more type of insertion reaction that we will look at. That is the insertion of ethylene into a MH bond. Giving us M C R R C R R H. So this kind of product is obtained when the ethylene group essentially gets inserted into the MH bond over here. Now this reaction proceeds via a four-centered intermediate, that is the ethylene there is interaction between the MH bond and the ethylene pi bond over here. This essentially gives us a four-centered intermediate to finally give us the product, that is C C H. This is how we obtain the product. Now, what we can observe here, what we can recollect here is that the reaction is exactly, or the opposite reaction essentially releases an alkene from the MH species from a metal alkyl complex over here, and this is nothing but beta hydride elimination reaction. So we already learned about the reverse reaction, that is the beta hydride elimination reaction, where a beta hydrogen atom essentially gets attached to the metal center in a metal alkyl complex, and this is the primary mechanism through which metal alkyl complexes decompose essentially, and we have also learned in detail about the pathways through which we can block this beta hydride elimination in metal alkyl complexes, and that is how we can stop the decomposition of metal alkyl complexes. And similarly, we can do the same sort of things to block to achieve the forward reaction as well. So this is how this insertion of olefin into MH bond essentially takes place. So with this, let me conclude this lecture. What we have learned during this lecture are about two different types of reaction, that is insertion reaction and elimination reaction, and we primarily looked at one type of insertion reaction, that is CO insertion reaction into a metal to carbon bond, and we looked at the mechanistic evidences that were available by using tagged carbon monoxide ligand in the one of the reaction with manganese carbonyl alkyl complex of tagged carbon monoxide. And what these evidences suggest is that instead of CO ligand getting inserted into a metal carbon bond, the alkyl groups migrate to one of the carbonyl ligand, and that is how the product is formed. So instead of terming the reactions as CO insertion reaction, we should term them as alkyl migration reaction. And then finally, we also looked at the insertion of olefin into a metal hydride bond, and this essentially brings us to the end of this lecture, and I Thank you very much for being with me during this lecture.